I think that, okay, like, long time ago. Yeah. Yeah, just the elevator across the hall. Yes.
The green is online. Okay.
Green is working, yes.
Yeah.
Okay.
Do you have any
got it,
pointer or take or?
I don't think we have like a physical pointer.
Oh, thank you.
Should work, just try.
Oh, thank you.
If it doesn't, there's a way to do the. You can come on like a little, this kind of thing. Oh, okay, that's okay.
Okay.
Yeah, maybe a little bit more, yeah, a pain to do, because you have to do it while looking at this computer, it's happening on the screen.
The next.
I'm going to present, check the presentation. Yeah, of course. This is it. My good presentation. Movies are starting. Oh, that's fine. Never know it. Okay. All right, up and here. Yeah, go. No, okay, no. And
Yeah. Ah, yeah, Sean is not available. Yeah, his travel budget was not approved time. Yeah, I'm Hiroshi Otomo.
Yeah.
The third one.
You're Otomo.
Yeah.
Okay.
Yeah.
Okay. You want me to walking in while you're trying to? Yeah. Otherwise, everyone is saying. It's the last session.
Yeah. Right.
But we could also push it back 5 minutes, so
Yeah.
I think that's at your discretion.
That's okay. I don't want to be expelled from Switzerland to take it back.
I think you get a warning the first time.
Yeah, a written letter.
Yes. You have to go before a judge and apologize. That's what happens if you are on the train without the right pass for the third time, you have to go to a judge and apologize.
Yeah. I didn't.
Based in the US, so.
Yes, yeah.
And that's a prototype.
No, actually, I'm a third one, Hiroshi Otomo.
Well,
Yeah. My origin is Japan, but I'm living in US.
I have friends with them.
Yes. Cool.
Yeah. You like Boston?
Yes, yeah.
Okay, I think we can start with after the welcome session on soft method physics. Our first speaker is Hiroshi Otomo from the US. And
Yeah, thank you so much for introducing me. So I'm talking about capillary flow simulation with the phase field lattice Boltzmann model. So with the recently developed phase field model, phase field lattice Boltzmann model for solving the Allen-Kahn equation was proposed 2016. And then after that, it is applied for various industrial cases, and then it can pass a lot of benchmark test cases. So it's very robust to solve the high density ratio flow. And then especially compared to the Kahn-Hilliard model, it can persist a small droplet. So it's a critical radius is small. And then so that's why we recently are using this model. So the model, the lattice Boltzmann model itself is written this way. So it's based on the original paper in 2016. So in this talk, on top of this model, we try to implement the proper wettability and friction model to simulate the accurate capillary flow. So the hydrodynamics side, we still keep using the original paper's formulation, except for some difference. The one main difference is the filter collision operator to stabilize the model, and also with some high order correction. So the more detail of the formulation can be seen in the published paper here. So on top of this physical lattice Boltzmann model, we try to think about a wettability model. So the naively, we implement this model on top of our surface technology. So our surface technology, we extend some sampling box from surface like this blue box, and then get the sampled value using this extended box. So the next wettability model. So the main key point is that when we compute the gradient phi, Laplacian phi, we have to refer the neighboring site. And then if that neighboring site is inside the solid, we have to deduce some value for this phi SI. So what we did is that naively, so we know that the surface normal for this surface. So rotating this n by assign the contact angle theta, then we can know how the gradient phi direction. So this dot, the perpendicular to this dotted line, the water profile should be varied. The next question is most of a phase field model, so profile should be hyperbolic tangent. So the next question is where is the tangent hyperbolic center? So because we know the sample value, for example, this yellow point, we know the value. So, and then we know the assumed hyperbolic tangent curve that varied from 0 to 1. So that's why we know the where the hyperbolic tangent center. So like this hyperbolic tangent profile along this line. And then with computing the effective distance by taking norm between W and then the CI, we can know the effective distance. And then with we just change the distance on top of a hyperbolic tangent profile. Then we can reduce phi SI. That's our naive approach here. So first test case is a slug between flat plates. So we put the high density component on the center, and then we with the density ratio of the 1,000, and then we tried various parameter options. And especially because we care about the lattice condition very much, so we changed the lattice offset by this magnitude. And then first result looks like this. So the qualitatively looks fine. So all the parameter is expected, and then pressure is reasonable, and sum of spurious current will be observed. Summary result here, the measurement, the input contact angle versus measured contact angle theta. So they seem to be consistent well. And then spurious current, so sorry, different color shows the different offset option. So they are crafted very well in this measured theta. And then maximum spurious current is a reasonable small. And then also for this always wettability model, especially for the very wetting case, we likely have like a film along the wall. But with this model, we monitor the density in the edge of domain. So we don't see much film here. So I think that's good things. And then next we move to the dynamic case. So we put the water in the center of the channel, long channel, and then it's pushed by air. So air is injected from left edge here. So here's a physical quantity. And then we know we have some insights based on the theory. For example, so here the main topic is to measure the pressure difference from left edge to right edge. So how it look like? So that is governed by firstly water viscous force, the first part. So assuming the positive flow, the analytical solution can be written like this way. So that's based on the previous study. And then second element is capillary pressure. That's a balanced capillary force between receding side and the advancing side. And then the contact angle should be different between receding and advancing. That difference is based on Cox-Boylan flow here. And the last one is wedge dissipation. So, but because of the distorted streamline around the interface, so interface have some excessive diffusion effect. So that's this term. So they also based on previous study. So with this theoretical value, we compare the simulation result. So this is the result. So unfortunately, we overestimated a lot. So it's a 23% overestimated, especially receding side, it's overestimated a lot compared to the theory. So this I think if we increase the number of slabs, this situation gets worse and worse. So we would like to improve this case. So firstly, we just come back some artificial. So here's a diffusive interface model. Interface is a kind of artificial thing. So that's definitely much thicker than the real one. So, but we have to treat some in some good way. So we firstly detect the interface region, and then we apply some wall model to calculate the friction. And then in that at that time, the molecular viscosity used for wall model is computed by mixture viscosity, and then we change this formula so that the water viscosity is used as much as possible. And also friction is reduced at only very limited interface region. And also hysteresis model is very simply implemented like in addition to the phi SI mentioned in previous slide, we just blend the phi SI computed on the gradient phi at the previous time step. So with this method, we take into account like a history effect. So the result here, so compared to the previous, the before the modification, this modification changes the receding side pressure very well, and then lastly, deviation become 3% range. So that seems to be promising. However, we have to pass all other cases based on this model. So that's challenging, including like a static case. So firstly, this is a critical pressure case at a static. So we have two different channel heights here, and then put the slug in the center. Because of different height, there's critical pressure from right to left, and then we apply the pressure difference from left to right to balance the capillary force. And then we ramp up the inlet pressure, and then see when slug start moving. So in that way, we compute the capillary pressure, how much is accurate. So we know the analytical solution, and then we use these parameters. Because it's 2D, it's easy to change the parameter. Yeah, so then we apply the 5% less than the analytical solution, 10% more than the analytical solution. Then we estimate how much the deviation can be, so that this summary table. So first column shows the channel height H, and the second column shows the contact angle. So I think all cases are within 10% range. So that's, I think, a reasonable range. And then next is a droplet on the inclined wall. So in some model, due to the lack of isotropy, even without force, droplet can easily move up to the right or left. But with this model, it's very stable. Then if we, so these are two different cases, two different inclined wall angle. So if we pick up this this portion, and then here's a contact angle line, it seems to be same, because I think contact angle is same, just the inclined angle change. So yeah, so that's why I think the lattice condition independence is pretty good. So this is a well-known case. So we put the droplet on the circular cylinder, and then this is a parameters. And so we know the analytical solution based on this paper. So we pretty much good agreement with the analytical solution. So also this is a 3D critical pressure measurement. So based on the 3D sinusoidal channel, we put the oil in the center, and then see how much the critical pressure is as a similar way as like a 2D case shown 2 or 3 page. Yeah. So we use this model parameters. Sorry, no, initial slug position, the condition parameters, contact angle, and then apply the 10% less and then 10% more than the analytical solution, then that's as we expected, the start slug moving. So this is the animation. So this is just beginning. So now it's a contact angle. It can be seen here. So I think this is 90 degrees. So, and then after some pressure difference, the drag start moving. It's called highness jump. So it's good. So last case is a dynamic case, so which is very challenging. So as previous cases, we have an air inlet from bottom, the left corner, and in the middle, water is injected. And then we measure the pressure difference between left edge to right edge. So this is a long channel, 96 millimeter. And we vary the water superficial velocity here while keeping the air super velocity. So we have the, so here in this previous study, there's an experimental result. So we compare the experimental result. So they tried two different wettability conditions. One is a whole wall hydrophobic, and the other one is a hydrophilic, except for the bottom wall. So bottom wall is hydrophobic. So this is the result. So the solid curve shows the experimental results, and then the red and blue circle shows the simulation result, hydrophilic and hydrophobic. They seem to be matched very well. So actually, with that, the recent modification, the pressure difference completely overestimated. So we definitely need this kind of a reduced friction treatment. And so here the y-axis is the pressure difference, and then x-axis is the water inject velocity. And then lastly, we also check the flow pattern. So the hydrophobic case, the most water inject case, we have a long slug, and then other side has like asymmetric small droplet. So that is seem to be consistent with their experimental phase diagram. And also the hydrophilic case, we have a film here. That is also consistent with the experiment, stratified flow. Yeah, so yeah, this is summary. So yeah, thank you so much.
When you're speaking about the hysteresis in your slope calculation, can you just briefly explain how you treat the hysteresis and also why you have a hysteresis if we assume that the smooth side of surface, you assume that there is no difference between the advancing and the listening config angle, but
Yes. Yeah. So actually, our hysteresis model is kind of preliminary, so we don't want to push so much. But what we did is just include some history effect. We just refer the previous time step 5 to when we compute the phi SI. So that's it. So, but I think strictly speaking, we need to check the Cox-Bonner flow consistency. And so that's, I think, kind of dynamic advancing and receding contact angle. The static advancing and receding is a little bit tricky, but the dynamic one, we can check with Cox-Bonner flow. So we plan to seriously check at that viewpoint.
Actually, that was the question I was wondering, if you actually play with the viscosity at the interface, how much does that actually change the slip length in the Cox-Bonner flow? But it sounds like you haven't tried to do it.
Yeah, that's interesting. Yeah, that's we definitely need to check. Yeah, not check at this study.
Thank you very much.
Yeah, thanks.
Sorry. Yeah.
Okay, so our next speaker is A33 from Rome.
Okay. Thanks. Okay, thank you very much. Thank you for the introduction. So this talk, I will speak a little bit about some lattice Boltzmann simulation of the migration of these active droplets through constrictions. On the right, you see an image from our active droplet. I'll try to explain later how we model this. And on the left, an image from living cells that actually inspired this work. This is a work that has been funded by an ERC Advanced Grant, what by Sauro Suchi, and has been developed in collaboration with the list of authors here below. Does it work? Okay. Okay. Okay, thanks. So just to set a little bit the stage, say that under physiological conditions, often living cells navigate through very complex environments, and they often encounter pores, such as in this microfluidic experiments. And the typical size of the pore is much narrower than that of the cell. But in spite of these differences, in many cases, the crossing is successful, like you can see in this movie. And there's a lot of research going on because this process is interesting in, for example, wound healing, in tumor metastasis. It's very well known that tumor cells can navigate very far from the initial tumor site, and they often cross very narrow constrictions. From the modeling point of view, our idea has been to essentially build a minimal computational model, minimal because it should rely in principle on a minimal set of key parameters based on these active gel droplets, which are known to capture some aspect of real cells. So the idea is to probably understand a little bit more on the crossing of living cells, and on the other hand, to possibly study and understand a little bit more of how an artificial microswimmer would behave when crossing through constrictions. So there are some connection with biology, with engineering. Okay, so just to tell you what an active gel droplet is, probably the first experiment was made by the group of Zvonimir Dojic in Brandeis University, in which they essentially have a water and oil emulsion here, and they encapsulate within the water droplets, an active material. In particular, this is a microtubule kinesin, and the kinesin is the active element essentially propelling the droplets. The active material is absorbed at the interfaces, and to a sort of Marangoni effect, they move. There is a lot of bio research going on in experiments. There are several groups, producing, realizing these droplets. And on the modeling side, let's say that lattice Boltzmann, often combined with phase field theory, has been proven quite a reliable approach, framework to capture some aspect of the hydrodynamics of this active gel, of the cells, in particular of the active gel droplets. You see here, for example, the fluid flow of a three-dimensional polar active fluid droplets, which is kind of similar to what you could see in this fluid flow from experiments of a moving tumor cell. This is obtained through PIV techniques. So this is just to tell you that there is, active gel droplets are realized, and there is also study modeling of these systems. So let's see the connections between active gel droplets and cells. So we are a little bit more interested in cell mechanics. And generally, one can see in a simplified way the cell as a dense network of protein filaments, actin, microtubules, myosin, and kinesin. You see here some of them. But it's generally accepted that for cell motility, there are a couple of ingredients which are very important, and these are actin filaments and myosin. And so we'll see our droplets essentially made in this way. So the active droplet is a fluid droplet containing these two ingredients, actin filaments and myosin. So we don't care of any other biochemical effects going on, or any other, let's say, protein. For example, there may be microtubules that could be important, and also kinesin. This is, for example, a simulations made by more or less 10 years ago by the group of Mike Case, Davide Maranduso, and Al Santillon when they were in Edinburgh, which they simulate the swimming of a three-dimensional droplets using exactly this model. So fluid droplets, actin filaments, and myosin. Okay, this has been done. Our idea is to extend this model to study their effect under constriction. So let's see how we model actin filaments and myosin, at least their effect at the mesoscale level, okay? So actin filaments, essentially, if one look closely at the structure of a cell, they look more or less oriented along a common direction, and they often display liquid crystal ordering properties. So typically one can either associate a nematic order to a nematic tensor or a polar order. We use the polar description. The polar order is typically found when actin filaments propel along the direction of motion. This happens, for example, in crawling cells when there is a process called actin polymerization. So the actin filaments are described by a liquid crystal with a polar order in this model. And then there is the myosin. The myosin is, the role of the myosin is essentially explained here. So the myosin typically pulls two actin filaments along opposite directions, essentially collecting the flow. Doesn't work anymore. Anyway, collecting the flow, let's say longitudinally, and expelling it perpendicularly to the actin filaments. And this can be modeled as two forces, in this way, creating this dipolar fluid flow structure. And if one, sum all over contributions of the actin filaments and myosin and coarse grain, then you get finally an expression of the contractile stress, which depends on the polarization here, and this parameter, which is called activity, which measures the strength on the active stress. This was obtained by Sriram Ramaswamy at the beginning of the 20s in a couple of PRL papers. So now let's see how we model this active gel droplet within a microchannel and how we model the constriction. So essentially, one has to define that set of hydrodynamic phase field, phi, which is essentially a order parameter, velocity of the fluid, polarization of the liquid crystal, and density of the fluid. So the droplet is described by a phase field that phi3, for example, which is that yellow region is different from zero inside and zero outside everywhere in the microchannel. This is a portion of the microchannel where there are flat walls on top and bottom. Then you have polarization just inside the droplet because we want liquid crystal and the actin filaments just inside. And then the pillars making a constriction are modeled as a static phase field. So essentially, these are other two phase fields, different from zero inside and zero outside, but essentially they don't evolve, they are frozen, okay? And the velocity of the fluid is zero inside in order to model it a bit more closely a solid object. As you can imagine, for people working a lot in lattice Boltzmann, this is a very rough and approximate modelization of a solid object, but despite of that, it's very effective in modeling constrictions, at least in this experiments, and modeling also adhesive effect, which, as I show later, are pretty important for the crossing. And then we have also the boundary conditions. In particular, we set neutral wetting for the pillars because we want the interface being perpendicular. We set no wetting for the droplets because we don't want the droplets being connected to the flat walls. And we have no slip condition essentially for the velocity field, which is very common in the microfluidics. Now, what are the evolution equation of this aerodynamic fields? This is, I will be very quick here. This is a very well-established framework. It's been discussed in many presentations. So essentially, you have the first two equations, which are advection relaxation equation for different phase field and for the liquid crystal, and then you have continuity and Navier-Stokes. The important ingredients are the active stress, that this guy here, the green circle, which is multiplied by phi3 because we want just the activity being inside, the droplet. And this equation also using a hybrid free energy lattice Boltzmann. The interesting fact is that, as I said before, the pillars phi1 and phi2 are essentially static. So what we do, we essentially start with a semicircular region, which the phi1 and phi2 are defined. We relax a little bit to smooth a little bit out the interface, and then we stop the evolution. Okay, so just to create this solid object, and the velocity is zero inside. To completely solve this equation, we need to define a free energy. Here, this capital F appearing also in the chemical potential. And for the people who are familiar with this formalism, this is an extension of the Landau-Dejean free energy. So there is the binary fluid contribution, set in surface tension and interfacial weight of the droplet. Then you have the liquid crystal contribution, where we have also the distortion. In particular, this is a splay deformation, one of the typical deformation of distortion of liquid crystals. And then we also have the repulsion contribution and the adhesion. The adhesion is the, let's say, important ingredient here. The addition essentially favors adhesive effect between droplets and pillars, and we model in a very simplified way. So essentially, there is the entry, I would say, of the pillar, half of the pillar, in which there is an adhesive coefficient, which is different from the one at the exit, okay? And in particular, the one at the entry has to be larger than that of the exit to observe within some particular ranges a successful crossing when the gap is, let's say, narrow. These adhesive effecting we also realized in experiments using particular fibronectin, and this has been done in some experiments. So let's say how, let's say, a droplet acquires motion, let's say in the bulk, so without any, without being confined, and then we see how it behaves. 2 minutes, okay, so I'll be very quick. How it behaves when it goes through constriction. So essentially, we start with the two-dimensional droplets in which we have a polarization uniform, and then we switch on the activity. When the activity is switched on, the droplet essentially elongates and doesn't move, essentially because you have this dipolar flow field in the surrounding. But then if the activity is sufficiently high, larger than the elastic deformation, there is this spontaneous symmetry breaking that creates plate deformation and two vortexes inside the droplet that propels the motion. This is a pretty well-established mechanism worked out more or less 10 years ago in the paper by Elsent-Jung and Michael Cates and Marenduzzo that explained how, let's say, these droplets can acquire motion just by the effect of the myosin. So what happens if you have a larger gap? With a larger gap, essentially the crossing is very easy. Okay, let me see if I can. I cannot use the. Okay. Anyway, that is the move. It takes a while. Just have a look at these pictures here. So the crossing would occur in this way, and then in this case, you don't have additions because the sides of the let's say the constriction is, say, comparable with that of the droplets, and there are just weak deformation of liquid crystals. So display distortion more or less are just remain there. If you look at the speed of the center of mass, there is just a decrease of speed when the droplets touches the constrictions, and then an increase, a sort of pushing out effect when the droplets comes out, and these are also captured by the experiments. If we go to smaller gaps, in this case, the situation, of course, is a bit more complex. What we see is that the crossing is successful if we have adhesive effect in the way I mentioned before, and in particular the adhesion. This is sort of an adhesion number, dimensional number, has to be larger than 1. So there should be stronger adhesion at the entrance because the droplet has to be connected with the pores, and lower adhesion at the exit because we want to have sufficient energy for droplets to kind of sneak out from the pore. We also have a decrease of speed when the droplets touches the constriction, and a couple of increase of speeds when the droplets squeeze in the pore and then comes out. And these are very similar to what has been observed experimentally. And we also have some, these blue arrows are band deformations, which can be observed locally and temporarily in this in constriction. So this is the case when the droplets simply doesn't cross because there is no addition. I can skip this one. This is a typical flow field, which is basically two vortexes out of the droplet, a single vortex plus a unidirectional flow field inside. And because the flow field is slightly larger at contact between interfaces and pillars, because that's the effect of the addition, providing the additional energy. And this is what happens when let's say the gap is very narrow. It's very similar to what you have seen before, except that there are larger shape deformations with stronger band distortions, which last longer. And some of these shapes, in particular, this is called hourglass shape and ampoule shapes, have been also observed experimentally. Okay. I skipped this one. This is just a calculation of the transmigration time, which is in, let's say, in agreement with what you can see experimentally. We compute the transmigration time of the order of hours, whereas real cells have a few hours more. That's just because in real cells you have, of course, the nucleus, which is a limiting factor for the crossing. You have also different viscosities. Our model is much simpler, so there is, for example, a single viscosity. Just one thing about the perspective, we are trying to possibly using machine learning technique to track, this active droplets to predict shapes, in particular, when there is situations which there is a dense emulsion, resembling a little bit tissues in microfluidic channel. Okay, sorry for being a little bit over time, but thank you very much.
Could you extend those to compressible flows?
Sorry, could you?
Extend.
Let's say the microfluidics, they generally, the fluid is uncompressible.
For like embryonization or morphology, right? If
Mhm.
like a tissue with extends, but you also have like a myosin field, right? Like an embryo form, so you
Mhm.
mix it to tape and then it starts to form the embryo, right? And like
Uh-huh.
a harder one. But to use that technique, so I was just wondering if you could extend it to compressed individuals because they need two-dimensional, they need compression.
But in theory, probably yes, but to have a compressible flow, and so I had to think a little bit about for compressible flow, what do you mean exactly? So, because I don't have a, yes, okay, that's okay, that's good, but we never generally work with compressible flows in this kind of experiment, so that's why. Okay, thanks.
So droplets pass through the constriction.
Mhm.
Probably there are some cases in which they don't pass.
Yeah, there are some cases which there's no crossing.
Or is condition to pass?
Yes, a little bit. So this was a couple of moves I kind of skipped. So essentially, if the gap is very narrow, you need addition. If there is no addition, it's very difficult to observe the crossing. So if you can also increase the speed, probably one can imagine that with more energy can cross it, but it doesn't happen. And also if you, for example, model the addition, saying that the addition coefficients at the enter and the x are the same values. Okay, if they are same values, there is no crossing in that case. So essentially, because they can get stuck in the pore without any possibility of going out. So there are cases in which the crossing is not successful. The shorter is the gap, the more difficult is the crossing, and the narrower is the regimes of some numbers to observe, this crossing. But in particular, let's say that in this model, the addition was found to be fundamental for observing the crossing.
So yeah. So I'm wondering if the morphologies look very different, whether you have active propulsion, which is your case, or if it's just a pressure difference between.
There might be actually. Because in particular, if I imagine experiments which we have espresso-driven flow, you can have, for example, a projectile, drop at that shape. Yeah, there might be differences. Although, I mean, of course, in real experiments, generally the droplets move in water, for example, in microfluidic channel with water, and they're self-propelled. So, of course, these shapes partially recover what you see in cells. Cells can have also a larger shape deformation with. But
my question is, if the shape that you see is just the fact that it has to pass through constriction one way or the other, or there is something that is intrinsically active that means the shape is changing in some way.
No, that's due to the constriction, because I mean, if you don't have the constriction, the shapes remains essentially that sort of spherical droplets going on. But then as effect of the constriction, probably a little bit of the active stress plays some role, and also the surface tension. One should compute also that. Be careful about the surface tension. But the resulting shape is, I mean, what you see when it crosses the constriction depends very much on the way you model, the constriction and the fact that it interacts in that way.
Good. So let's take our speaker here.
Okay. Yes. Mhm. Yeah. So, good afternoon, everyone, and thank you for the organization for letting me present this work. And so this is, it's good that Adriano presented before me, but it's a similar system. And it's also related to what Inácio presented yesterday or Tuesday, right? About active turbulence, and related to what Nuno will present tomorrow. So
I invite you to go. So this was done in collaboration with some experimentalists in the University of Barcelona, which confirmed some findings from sorry. From the simulations. Thank you. Okay, so recalling what is active nematics is a kind of anisotropic fluid made of elongated particles that tend to align to the neighbors. So and those particles, they are able to self-propel. So one example, it's colonies of bacteria, as you can see this in this image. So if you take a small piece of this image, you see that most of the particles, they are aligned with the neighbors. Another example are epithelial cells. They are usually isotropic, but when they are flowing, they become more elongated and they align with the neighbor and form those very typical topological defects that are observed in active nematics. So, but the kind of system that I will be modeling are mixtures of microtubule and kinesin. So those are two components of the cells. The microtubule, they give the rigidity of the cells and form the flagella of the bacteria, for instance, while the kinesin, they are those micromotors that transport organelles from one side to the other, and they use the microtubule as guides. So someone has a very good idea to mix those two components in an oil-water interface, so they sediment to this interface, so that there is little friction. And those molecular motors, they made those the microtubule to slide one related to the other, like it is illustrated in this image. And when you have many of them, you have what is called active turbulence. You have a system like this that is chaotic. It resembles fluid turbulence, but it is in a very small scale, in the micrometer scale. So very low Reynolds number. It's not inertial turbulence as we are used to in normal fluids. So how do we model this? So we use a model called Beres-Edwards model. It's a continuum description. So we have all those elongated particles. You don't consider them individually. So we consider averages. It's a mean field theory. And the orientation of those particles, they are encoded in this tensor order parameter. The alignment of those particles, they interact with the gradients of the velocity field. So they, depending on the kind of the particle, they tend to align or not with the gradient. There is a penalization for particles that are not aligned with the neighbor. So there is an energy cost. If you don't have forces acting, all the particles will be pointing in the same direction. And most important, there is an active stress that gives the motility of the particles. So, so in the equations, the first one gives the time evolution of the tensor or the parameter, so the alignment of the particles. And this I solve using finite differences. So I do implement as it is. There are more terms that I don't show here, like this S. But note that it depends on the velocity field, and the evolution of the velocity field is given by the Navier-Stokes equation with a model using Letzi-Boltzmann. So the Navier-Stokes equation is basically the usual one for Newtonian fluids, but it includes here the active stress. This part of the equation is the standard LBM that I use to model. So D2Q9 for 2 dimensions, second order expansion, BGK for the collision operator. And this term I include using as a force, so using Google forcing scheme. For those that I use it to simulate, so this is a system of 2 coupled equations. One provides the tensor order parameter and the other the velocity field. And as I was telling, those that are used to simulate active kinematics, we notice that there is a term that is missing here, that is the passive stress for liquid crystals. But it was found recently in experiment that this term is 2 orders of magnitude smaller than the active stress. So we can neglect this term. We also measured that, and even in simulations, it's quite small. So since it's quite complicated, there is no need to include it. Okay, so what do we do with this system? There are some proposals to use this system, this mixture of microtubule kinesin, in microfluidic setups. So, but if you want to build those setups, you need some pumps. But remember that the system is 2-dimensional, embedded in a 3-dimensional fluid. So how do we pump fluid? Well, we tried several setups, and we found that a regular lattice of triangles is able to direct the flow. So we can use this as a pump in a microfluid setup like this. So this is a simulation. We show here the director field. I used here a filtering paraview called the line integral convolution that shows the director field in this way. And I show in this way because it's quite similar to the experiment. So we asked the experiment in Barcelona because they invented a technique to solidify parts of the active kinematic. So they shed light in a certain region of the active kinematic, and this part becomes solid. So it can become a flexible solid or a rigid, depends on the intensity of the light. So we see here the formation of a capsule, something like this, that is quite flexible. But if they change the properties of the light, it will become more rigid. And that's what we need to reproduce this system experimentally. So they have initially the turbulent system or chaotic system, as you prefer, and then they shed light, and then there is the formation of those lanes that direct the flow in the middle of those channels, as in the simulation. So we somehow confirmed what we predicted using the simulations. And here there is a comparison for directed field, both in experiment and simulations. The velocity field, both systems, we see that this is an average in time. So we see that the velocity is mostly pointing to the right in the middle of those channels. And there are some red spots. So if you take the velocity profile in this line, we see some peaks behind each obstacle. And we also see the formation of some vortices, some localized vortices behind each triangle, both in experiment and simulation. And when we plot the velocity field, we see clearly those localized vortices. So there are 2 for each triangle. And we believe that this helps the rectification of the flow. So there are 2 vortices here that are continually pumping fluid to the right. Okay, so we characterized this system. We did some optimization, or for instance, we kept constant the distance between the defects and changing only the size of the obstacle, only the size of the obstacles, this h. And we see that there is an initial regime in which when we increase the size of the triangle, it pumps more, so the velocity gets higher, and then it reaches a maximum. So this maximum occur when the size of the obstacle is comparable to the average distance between defects. So those defects that are moving in the fluid, they have a typical distance, and it makes sense that there is a maximum for this point. So we also changed the separation between defects, so this is an optimization. Another interesting thing is that to pump better the fluid, we need a regular lattice. So one thing that we did, we displaced each triangle using a random displacement following a Gaussian, and this Gaussian has a standard deviation sigma. So we see here for a small sigma, intermediate, and high sigma, which becomes quite chaotic. And when we measure the velocity against this randomness, we see that the average, the velocity decays, and it should eventually go to 0 when the system becomes random. So it's important that the system is quite regular. So and in the experiment, it's more difficult to guarantee that it's regular. So it might explain some small difference that we observe. Okay, and we also compared this kind of system, or we made an analogy of the system with a combination of pumps. So so for instance, here we have pumps, something analogous to pumps in series. We have this unit cell is analogous to one pump. We keep constant the number of lines and change the number of columns. And this is analogous to plugging many pumps in series. And then we see here the velocity and pressure profile. The pressure is something that the experiment cannot measure, so it's something that the simulations complement the experiment. And when we plot the velocity against the velocity and the pressure against the number of pumps in the horizontal, we see that there is a regime in which increase, and then it saturates. Above a certain number of pumps in series, the velocity gets constant, and the pressure keeps increasing almost linearly. So this regime, this last regime, is what we would expect for ideal hydraulic pumps. So if you keep adding pumps in series, we expect that the pressure drop increases, but the flow rate remains constant. So this is analogous to pumps in series, and also compared for pumps in parallel. So now we change the number of pumps in the vertical, and we see that the flow rate increases linearly, while the pressure is almost constant. This is also the behavior expected for ideal hydraulic pumps. So this there is a good analogy between this system and hydraulic pumps combined. Okay, and well, if you have pumps, you would like to pump fluid in some geometry. So here we illustrate pumping fluid into a channel. We also tried with lattice of circles, but I don't show here. So both in experiment and in simulations, observe that it's pumping fluid. For instance, in this plot, we see the average velocity inside the channel for different separation between the plates. And without the pump, the velocity inside the channel is roughly 0, while with the pump is always positive, as we would expect. It shall always go to the right. So it's always pumping. And we can have more complicated microfluidic setups, like in this big vortex. You see here, we see the trajectories of the tracers that they do follow the expected path described by the triangle. So we could play with that in microfluidic setups, and they also did that in the experiment. Okay. So, and another application that you could imagine for this system is in micromixing. So, because mixing fluids at the micro scale is complicated because of the Stokes regime. It's completely reversible, right? So one strategy used in microfluidics is to use these tortuous flows, but you could also use this kind of system. So if you use a more isotropic system like a lattice of circles, the system do mix the solid. If you have these two droplets with different colors, here they are tracers, but we see that they do mix. However, the center of mass remains constant. So this is not very practical for applications. So we would put two droplets and they would mix, and that's it. It's finished. But we would like to keep injecting the fluid. So in this lattice of triangles, we see that the two droplets do mix because there is flow, transversal flow in the vertical as well. But the solute, the mixed solute, keeps going to the right. So that's what we'd like to have in applications, something like this. We'd like to inject two fluids in one side, they mix in the middle, and in the end you have the mixed solute, and you collect that. So this is better for this kind of application. So we see here the histogram of the velocities of those tracers. Both in experiment and simulations, most of them go to the right, and there is some distribution. Have measured the time that each tracer remains in the same channel, and this the time that the probability of remaining in the same channel decays exponentially with the duration. So there is a larger probability that they stay very shortly. That's the conclusion. And it's very unlikely that they remain on the same channel. So this is good for mix. Okay, and so to conclude, we have studied a system that is there are possible applications in microfluidic setups using this mixture of kinase in microtubule. We have studied both using experiment and in simulations. Let's boot and find different simulations. It's a hybrid scheme. And we showed how to use this in mixing applications. And that's it. Thank you for your attention. No, well, because well, I have here 3,000 for each color. The system is quite large. So each unit cell here is 128. And yeah, you have 6 by 30. So in the end, they become quite dispersed. Well, to study mixing of solid, maybe the better would be to use continuum theory, like a true solid, some distribution, some concentration of solids. But no, I could add. It just takes longer if you add more, because you have to calculate the trajectories. That's it. Yeah. Okay. Well, in the experiment, they do have discrete tracer as well. So one would be, but well, in the experiment, they could have one in simulation as well. But if you want statistics, well, don't know. I have many. Yeah. Ideally infinite. Yeah.
Have you guys looked at how this translates into 3D geometries?
3D geometries, good question. No, have not looked at that into that. That's also interesting. Well, if you have, you need something like a prism, like to direct the flow. That could be a continuation for this study. But I think in the experiment is way more complicated to do in 3D, because in the simulations, no, the model is, well, is just changing the LEDs from the 2Q9 from the 3Q918. But in the experiment, it is more complicated, because they usually use this interface, the sediment or the plate to this interface between oil and water, and the 2D system is quite clear to see. In 3D, it's possible. The Dodge group have done that, but it's way more complicated. But yeah, it's possible in principle. Yeah. But we have not looked at it.
So let's thank our speaker.
Sure.
One second. Still telling.